really thank for giving me the opportunity to present my work here. So I'm a computational molecular biophysicist, and one of the major research in our lab is to understand the transport phenomenon of proteins on DNA. So as all of us know that the processes like this transcription, damage repair or replication, and even this gene editing, which is currently a hot topic after this 2020 Nobel Prize. So the common factor in all of these processes is a specific binding of a relevant protein molecule at a target DNA site. So definitely this specific binding is important, but as some says that journey is often more beautiful than the destination, we try to investigate the journey. And so if you look into the nucleosome structure and the DNA conformations, this is quite messy inside nucleosome, inside nucleus. And I found it interesting that there is an analogy with the road transport. So for example, if you see one person, let's say, would like to come from New Delhi station to JNU, and you can have actually multiple ways to commute. So one can take a cab, one can take an auto rickshaw, or maybe metro, or maybe combination of all these, right? So even we find that inside cell, protein also takes multiple routes, multiple ways to commute, multiple modes to commute. And among those, one of the most exciting and interesting mode is sliding dynamics. So what happens is that this protein nicely rotates around the DNA major grooves while it moves on the DNA. So the result is you get this kind of spiral pathway around the DNA major grooves. And in fact, this kind of movement was experimentally determined by or experimentally verified by some groups. And one of that is by Sunny Z group, where Professor Bhattacherjee was also there, who nicely nicely described the diffusion of this protein for this spiral kind of movement. So we, along with some other groups, are continuously working in this direction for past several years, and we are trying to find out the details of such motions. So another one is that protein, like we do, protein also takes shortcut to scan its target site. So for example, if a protein is multimeric or it has a disordered tails which can interact with the DNA segment, so the protein, when two DNA sites are close to each other, the protein can really make a jump from one DNA strand or one DNA stretch to another, and thereby bypass a huge scanning of huge length. So that is kind of advantageous for it to facilitate or speed up its search process. We call this as intersegmental transfer. And obviously one cannot bypass the traffic. So like on road we have, in nucleus also you have many proteins which are like Crowder molecules, and they can prevent the diffusion presumably of a protein. So we looked into this quite a bit, this aspect, and we found that in presence of these Crowder molecules, a depletion layer forms around the DNA. And the Crowder molecules are not allowed to enter into this depletion layer. But the interesting fact is that when the protein is within this depletion layer, it experiences a microviscosity. Definitely the microviscosity is much lower than the bulk solution, and therefore when the protein is near the DNA, it can really move faster compared to when it is in bulk. And we have classified many aspects of this. We looked into, for example, the formation of this depletion layer, and we found that the width of this depletion layer decreases with the increasing Crowder concentration. So the size, shape, and the geometry of the Crowder molecules also impact the search process. And we also find that the interaction with these Crowder molecules are really very important. And often that interaction facilitates the search process of the protein to its target site. So another obvious, and this is the last but not the least, that as we have traffic signals on DNA also, what happens is that, let's say this red star is the target site where the protein needs to, this ball is the protein, it needs to bind. So there are many sites on DNA which are quite close to this target site, but not exactly. But protein may typically makes a mistake because these sites are very closely resemble with the target site. The protein may bind with these sites and stay there for some time. So basically, this kind of non-specific stability or affinity at sites slows down its search process. And this gives rise to a fantastic paradox, which is called speed stability paradox, which says that if you really would like to increase the speed, you have to compromise in terms of affinity. So if you have affinity, then the speed is compromised. So recently we took a closer look onto this, and we looked into how protein searches on nucleosomal DNA. So as you know that nucleosome can have a breathing dynamic, so it can partially wrap, unwrap with certain rate, and a protein can invade nucleosome when it is partially unwrapped. But in reality, we found that there is only a subclass of protein which can actually invade nucleosome, and we call this as pioneer factors. So we wanted to know what is so special in pioneer factor that can that enables it to invade nucleosome, whereas the other transcription factor cannot. So we constructed a generalized discrete state stochastic model for analyzing this target search process. So we, as different studies says that the pioneer factors can even bind with the nucleosomal DNA when it is in closed conformation. And in comparison, non-pioneer factor really cannot do that. It can bind with the DNA only when the DNA is partially unwrapped. And moreover, study says that the rewrapping of DNA or this kind of nucleosome breathing actually increase the dissociation rate of the non-pioneer factors from the nucleosome surface. So fortunately, we found some kinetic rates of two pioneer factors, RAB1 and CBF1, and one non-pioneer factors on nucleosome as well as on naked DNA or linear DNA. So interesting part is that we note here that the binding affinity is much higher for the pioneer factors compared to the non-pioneer factors. So we estimated the target search time for this kind of situation, and what we found is that, so the top panel shows the target search time on the nucleosomal DNA, whereas the lower is for naked or linear DNA. So what we found is that two major conclusions. One is that pioneer factors are way faster compared to the non-pioneer factors on nucleosomal DNA. The other way is true for naked DNA. The second thing is that the pioneer factors are fast, but their affinity is higher. So definitely it says that higher affinity is not really bad, which is completely in contradiction with what we have on linear DNA. So we tried to understand why it is so, and possibly we found out it is because maybe because of the nucleosome dynamics. So we tried to look into the impact of that. So what we found is that if nucleosome dynamics or kinetic rates, the opening rate, if you increase that, so what we get a drop in target search time. So which we find is because that when you increase the rate, when the nucleosome opens quickly, that means the waiting time is actually low. So as because the pioneer factors are associated already with the nucleosome surface, so as the waiting time is lower, so pioneer factor, as soon as it unwraps, pioneer factor starts is scanning. So it reaches the site faster. And we looked it for non-pioneer factors, and what we found is a fantastic U-shaped curve where we found that obviously when so this nucleosome opening rate is connected with its dissociation, because as study suggested, that is was our model. So what we found is that, so every time it dissociates, so if the dissociation is very high, the rate is very high, definitely it will take a long time. Whereas if the dissociation is very low, then also it takes quite a long time. And the reason is that when it performs a search, the non-pioneer factors, it may get stuck into some sites. And from that, it's very essential to get a restart or resetting. So this dissociation, nucleosome rewrapping induced dissociation, is actually a resetting mechanism. So we found that this resetting mechanism is actually helpful, and there is an optimal resetting rate for which you get the fastest target search. So we thought probably this will we should observe on the pioneer factor as well. So we looked into pioneer factor with respect to its dissociation from the nucleosome surface, and we found the similar kind of graph, but which suggests one thing is that there is an optimal error correction mechanism operates for this kind of system. Number one. Number two is that if you check this k-on rates, for as the k-on rate increases, the target search time or the resetting rate also increases. So possibly there is a correspondence between this on rate and the off rate. So we tried to find out that, and what we found is that there is a nice correspondence between off rate and on rate, and we found that when they are comparable, you get the fastest target search time. So that this red line shows the fastest target search time for this k on and k off values. So we came up with a new mechanism that says that there is a dissociation compensated association mechanism, which says that if as the pioneer, and if you see, this is the fastest search, and if you deviate much from it, basically you are making more errors. But let's say this black line is within 10% more time compared to this the fastest target search time. And we find that both the pioneer factors are within this limit, whereas the non-pioneer factor is much away. It's quite away. So the point is that for pioneer factors, its association is typically slow on nucleosome. But if the association is slow, then its dissociation compensates. Its dissociation is even slower, and it compensates that, okay, you take your time and search the DNA properly. But if association is faster, so to reach the target site faster, its dissociation needs to be higher, because if association is high, then there is a high propensity that the protein can get stuck into some sites which is unwanted. So in that case, this resetting mechanism works. So basically, there is a correspondence between these two rates which governs the search process on nucleosome. And what we find that this high binding affinity of proteins to DNA may not be a retarding factor. Rather, on nucleosomal DNA, it helps pioneer factor to minimize the impact of nucleosome dynamics. And the fastest transport to target site, target DNA site, is observed when nucleosome association and dissociation rates of a protein are comparable. The dissociation compensated mechanism ensures trade-off between nuclear mobility and minimizing the error in the search process. I will quickly speak about another transport process of a protein, but this time not on double-stranded DNA, but on single-stranded DNA. And as many of us know that single-stranded DNA are extremely vulnerable to chemical degradation. And there are certain proteins which kind of provide stability to single-stranded DNA. And RPA is one of the important eukaryotic protein which provides stability, and it is involved in many DNA metabolic processes. So the question here is that was quite interesting. So what we found is that the same RPA interacts with single-stranded DNA in both DNA repair and replication. What we found, the only difference is that the long and long ssDNA intermediate binds to RPA during DNA replication. In comparison, short ssDNA binds to RPA during the repairing the DNA damage. So our question is, what is so different in the length of the DNA that governs two completely different DNA metabolic processes? And also in from the literature says that this the long ssDNA, this is quite short-lived. So it has to bind very quickly, otherwise it will be degraded chemically. Whereas the short ssDNA, it forms a stable RPA DNA complex. So we try to understand this, and for that we developed a completely different model of ssDNA and RPA, ssDNA and protein, and we tested on it's completely unbiased. We tested it on various ssDNA protein complexes and successfully captured their binding. So we also did it on the RPA, and we found a fantastic structure which resembles completely with the crystal structure. Interestingly, people have also tried to look into this DNA level dependent dynamics on RPA. So what they tried is that they tried to crystallize ssDNA with RPA, where ssDNA is of a length one case in 25 base pair, 25 bases, and in one cases they have taken around 60 nucleotides. Interestingly, their crystal structure could only capture 25 nucleotide. The excess nucleotide are not captured. So basically, whatever they did, they actually produce the same crystal structure for both the length of the DNA. So then that actually tells us the problem is even more difficult when you do this because of the intrinsic flexibility of the ssDNA molecule. So what we did, we basically these are the two domains A and B, whereas these are the other domains C, D, and E. So what we found is that we divided this DNA into two halves. So one is the left arm, this arm we call it arm 1, and this is arm 2, and we monitored the formation of the contacts in these two arms. So what we found is that when you have a short DNA, this interacts with the protein and you find a fantastic minimum. This is the contact of arm 2 and contact of arm 1, a fantastic minimum, which is, we name it as H25. And it forms a nicely horseshoe-shaped U-like structure on the protein, which perfectly matches with the crystal structure. When you have long DNA, we find two minima. This minima, we find the protein, the DNAs are quite linear in shape. So basically the arm 1 contacts are already formed, high arm 1 contacts, but very low arm 2 contacts. In comparison, after some time, we got the final structure where we again got a U-like fantastic structure, this H62. So a couple of things. One is that we found that these two energy minima are separated by nominal free energy barrier. So that means it has a dynamic equilibrium between these two minima. Another one is that we found that this H25 is a bit more stable compared to H62, which is in accordance with the experimental study. And also this linear structure, this has never been discussed previously, and we have captured this for the first time, characterized it, this structure. And but there is an experiment which tried to find out what they have done. They have labeled the DNA at these two ends, CY3 and CY3 and CY5, and then they assumed if it forms a U-shaped structure, it should give a high signal in Fred. But what they indeed found is that the Fred signal actually decreases. So from that, they kind of assumed that probably these two ends go away from each other, but we really found this linear kind of structure. Next is we wanted to understand how this DNA moves on the protein. So what we found, this is a short DNA, and it moves nicely on the DNA. It attaches, and what we found is that this arm 1 is attached, and this arm 2 is now going through, diffuses slowly on the protein surface and coming this way. So we tried to find out the way, and what we found is that when the DNA binds with the protein,
you need to wind up.
Yeah, one more slide. Yes. So when it try to diffuse on the protein, it forms some sort of bulges, and these bulges forms and dissolute, and the DNA moves. When you have a small DNA, you can clearly see that this bulge actually moves. But for longer DNA, you see many static bulges, and the bulges are not dynamic at all. And in fact, in arm 2, you do not see any bulge formation. So we tried to understand what is going on, and what we found is that the association rate at this domain, this D domain, is independent of the DNA length, whereas the association at the A domain increases with the change in the DNA length, with the increase in the DNA length. So what we found is that when arm1 is, if you see the contact formation, arm1 contact forms and there is a sudden jump, which suggests there is a huge cooperativity in arm2 contact formation. So the arm1 contact formation facilitates basically the contact formation in the arm2. We measured the corresponding Hill coefficient and we see that with the increase in the length of the DNA, it increases linearly. So basically we found that this is a kind of fly casting mechanism which allows the diffusion higher, which explains the higher diffusion and explains why this long DNA can quickly bind to the protein. So in conclusion, I would say that the mechanism of binding to RPA by the ssDNA differs with the length of ssDNA. The short length binds through replication dynamics, whereas the long ssDNA binds in a cooperative fashion. The cooperative binding involves a conformational change from a stable linear intermediate to a horseshoe-shaped final state. And the presence of these two distinct binding modes are connected via dynamic equilibrium. So RPA associates more strongly with the short length ssDNA, but such process is much faster on the long ssDNA molecule. So I'll stop here, and so basically these work were done by two of these PhD students, and we are enjoying our journey on different track definitely. But thank you.